The overall goal of the following experiment is to accurately quantify biomarker expression across whole tissue sections of human cancer in order to assign a numerical value to the degree of heterogeneity in protein expression. This is achieved by first scanning immunofluorescently labeled sections of cancer tissue using a whole slide fluorescence scanner. Next, regions of interest within the tumor are selected for evaluation. Then, the expression of the protein of interest within the invasive tumor areas is quantified using the InHwa analysis automated image analysis system. Ultimately, this method can be used to map biomarker expression in cancer tissue, which is often very heterogeneous across the tissue section.
The main advantage of using these techniques over traditional methods such as immunohistochemistry using colorimetric visualization reagents is that immunofluorescence is more quantitative and more sensitive. Coupling these analytical methods with whole section immunofluorescence scanning enables fine-grained mapping of subtle variations in biomarker expression across tissues for the first time. Demonstrating these procedures is Mark Gustavson, a Director of Operations at the HistoRx laboratory.
After microtome sectioning of a cancer tissue biopsy and immunofluorescence staining of the specimen for expression of the biomarkers of interest, load up to 5 slides of the stained sections into the slide cassette of the slide scanner. Then, using the interface